If you printed out this handout that was posted, you can use this to help fill in the blanks as we go. The agenda for today is to start with 5 myths about perception that most people believe to be true, but in fact are not true. We're also going to talk about the 10 top features of the environment that perception cares about. And along the way, we're going to cover 13 classic and cutting-edge methods that are typically used in sensation and perception. So while taking notes, you can also fill out the sheet as a way to help guide you through the lecture. All right, so to start, we're going to discuss 5 myths that most people believe about perception. And the first myth is that you see everything. People have this impression that if something's in front of you and is visible, that we see it. And this is not exactly the case. And take, for example, a situation in which you were looking for something. Maybe you were looking for the ketchup bottle in the refrigerator. Maybe you were looking for your keys on your desk. And even though the item was right there in front of you, it might have taken a while to find it. And in fact, it's really frustrating because you think, it's right here. How could I not see this? All right, well, this provides an example of situations in which something's clearly visible. It's right in front of you, and yet you don't see it. In the lab, this is studied with a paradigm known as change blindness. And in change blindness, people are shown an image and then a second image. And everything in the second image is the same as the first image, except one thing is different. And the task is to find the difference. So let's do an example. Try to find the difference here. Raise your hand if you have found it. Okay, about half the class, a little less than half the class. Look at the white line. Oh, right, now you see it easily. The white line was in front of you the whole time. It was clearly visible, yet it takes some time to find it. And in fact, a third of the people in these studies never see it. They never find it. So the change blindness is one technique to help us understand that perception does not see everything all the time, but rather only sees a select few things. The next myth is that we see things as they really are in the environment. If there's something in the physical world, our perception of that thing is accurate. It's veridical. And this isn't quite true either. How many of you see motion in this image? Yeah, so a lot of people more towards the front. This image is completely stationary, yet most people report seeing some kind of motion in the image. This is an example of a visual illusion. Visual illusions are a great technique for studying perception because they show errors that the system makes. And in looking at these errors, we can get at some of the assumptions that the system makes about what is in the environment and what we see. The third myth is that we see what projects onto the back of the eye. So all incoming information, light, projects to the back of the eye, and that projects to a place known as the retina and creates this retina image. And a lot of people think that whatever projects to the back of the eye is what we see. But in fact, that's not the case. This is a famous illusion known as the Ponzo illusion. And most people report seeing this white bar as bigger than this white bar. But in fact, they're the exact same size. Meaning if you took a ruler and measured those white lines, they would measure to be the exact same size. Which means they're projecting to the exact same size on your retina. But that's not what you see. What you see relates to what's out in the world. Perception doesn't care what's on the back of your eye. Perception wants to know what's out there. And so you can have situations such as this illusion where what you see is different than what projects onto the back of the eye. The fourth myth is that perception is a single system. We talk about the perceptual system and what we perceive, but in fact, there are multiple systems. So, for example, this is another famous illusion known as the Ebbinghaus illusion. Most people see the orange circle on the right as bigger than the orange circle on the left. So our conscious perception is that one circle is bigger than the other. However, if the orange circles were discs, and you tried to reach and grasp one of them, you would open your fingers the exact same size for both circles. So even though you see the circle on the left is smaller, you would open your hand the exact same size as you would for the circle on the right. So there's one system responsible for our conscious perception. There's another system responsible for guiding our actions. And while conscious perception might be fooled by these illusions, thankfully our action system is not fooled. And it allows us to navigate through the world without making too many errors. And then the last myth is that perception's a passive process. And what I want you to do for me is I'm going to show you a bunch of shapes that are different colors, and I'm going to announce a color. And I want you to figure out what shape is the object that's that same color. All right, here we go. Red. Purple. Blue. Green. Great, good work. Did you find that you looked at the object to see its shape? Or did you look in the center of the screen without ever moving your eyes? How many people moved their eyes to the color I announced? Yeah, almost everybody, right? When you need to know information, you go and you get it. You go and you look for it. You don't just passively sit there and take it in. You actively go out and see it. Eye tracking movements are another measure that's often used in perception. So there's several devices that allow us to track where people are looking. And through this eye tracking, we can see what kinds of things in the environment attract your attention, attract your perception. And that's another method that's often used to study perception. All right, so here are different things that perception is not. Let's consider what perception is. And to do this, let's start with the question of why would you want a perceptual system in the first place? So here are two example critters. We have Iris and Nosy. And just like their names imply, Iris has a perceptual system and can detect things in the environment, and Nosy cannot. Nosy has no perceptual systems at all. How many of you would prefer to be Iris? Yeah, most of you, right? And why? Well, this question is getting at why is perception important? What is perception giving us that we can't get without perception? And one answer is that perception is our only link to the outside world. Without perception, you would be completely unaware and have no knowledge and no ability to detect anything in the environment. And without knowing what's in the environment, it's going to be really hard to survive. So the second reason why perception is important is that it's key to survival. So if we reframe this question, who is more likely to survive? Iris is more likely to survive. Now we know that perception is not just seeing everything. It's not a singular system. It's not a passive process. So what is it? And what it is it's extracting information about the environment that you need to survive. Perception is about extracting information about the environment that you need to survive. And so for the rest of class today, we're going to talk about these things that are extracted from the environment. And we're going to do a top 10 list. The top 10 things that we need to know about the environment in order to survive. And I want you to take a few minutes and guess. Guess what's on this list. So write down 3 guesses of things that you think are going to appear on this list. All right, so when I query my class, I teach the Psych 310 class on sensation and perception, and when I query my students in that class, one of the top things that always gets raised is motion. How many of you put down motion? Yeah, some of you. So motion is something that's really important to know about the environment. And rightly so, there are special areas in the brain that are dedicated just for perceiving motion. And this starts within the eye. So here's the eye. At the back of the eye is the retina. And the retina is a layer of 5 different types of neurons that process the light. And here's the retina here. The very last layer in the retina, before information gets sent to the brain, contains cells known as ganglion cells. And ganglion cells process specific features in the environment. And there are 2 types of ganglion cells. There are P cells and M cells. And P cells code for things like color and texture. And the M cells code only for motion. The M cells, M for motion, are a system that is what we use to see motion. If you lesioned your M cells, you would not be able to see motion at all. And if you didn't have any P cells, you would only see motion. So M cells are a system that allows us to see motion. From the eye, information from the M cells projects to the thalamus. And what you can see in this image of the thalamus is that there's different layers. There are 6 different layers. And the M cells project to the first 2 layers, and only the first 2 layers. So this information is segregated throughout its path to the brain. And there are dedicated areas in the brain and the thalamus, and then later pathways that are dedicated just for seeing motion. But not all motion is equal. There are certain kinds of motion that are more important to us and more important to surviving than other kinds of motions. And to give you an example, a projectile coming right at your head is a pretty important motion to be able to see. And in fact, there's a dedicated system just for that, and that's known as looming. So looming are objects that are coming right at you. And we're really good at seeing this. And so I want to give you a video that shows just how good we are. All right, here it is. All right, so what you may have noticed was just how good President Bush was at ducking out of the way of that shoe. All right, and that shoe came right at his head, and he reacted very fast. Watch it again, though, and watch the Prime Minister next to his side and see how he reacts. No reaction. The president's already out of the way, and the Prime Minister doesn't know what's happening. Is he less athletic? No, probably not. Rather, the shoe's not coming at his head. So the shoe has not triggered this special system that detects looming. All right? If something's coming at your head, that's going to trigger this system, and you can react. If something's not coming at your head, it's not going to trigger the system. You're not going to react nearly as fast. The way that this is studied in the lab, the task is to determine whether the oval was a vertical oval or a horizontal oval. All right, so here, and it's a hard task. So this is the oval. Is it vertical or horizontal? It's vertical. And then there are some circles up there. Those are known as distractors. And this task is known as a visual search task. It's one of the more powerful tools that perceptual psychologists use to study processes. And what we measure is the time that it takes to find the target. And the typical finding is that as you add more distractors, the time to find the target takes longer. So that's a typical visual search paradigm. Now, in this experiment, they added something different. Right before the target display was present, there was a cue. And the cue was a thing that came towards you. And the cue was presented either at the same location as the target or at the same location as a distractor. And the cue was either something coming right at your head, so it would trigger that looming system, or it was coming at you, but it was going to miss you. So it was just coming to your side. And what they found was that you were the fastest at detecting the target when the cue happened at the same location as the target and the cue was looming. If the cue was a near miss and at the same location as the target, you weren't nearly as fast. Right? So it's this square here, this combination of cues, that makes you the fastest. And the idea is that this is tapping into this looming system. In fact, this is one of the ways we know we have this looming system. All right, so looming is an important type of motion. Another important type of motion is going to be demonstrated next. So what do you see? Most people see a random array of dots, but as soon as it starts moving, what is it? It's a person. What's the person doing? They're kickboxing, they're playing karate, they're kicking a ball, they're running, they're jumping. All that is a few dots of light moving around. And yet, from that information, you can extract so much about the scene. Is the person male or female? How many think male? How many think female? Yeah, most people see male. Right? So the only information out there is the motion of the body. This is a technique known as point light walkers. And the way you create these is that you put these infrared sensors at various places around the body and use special cameras that can detect infrared light. So you're only recording these points of motion, and then you can create these videos. And what's so compelling about these videos is you can't help but see biological motion. And in fact, biological motion is a special kind of motion. It has its own rules. It follows its own set of principles, different from that which applies to regular motion or non-biological motion. All right, so for our top 10 list, we're going to put down motion. We're also going to put down looming as a separate item and biological motion as a separate item. The next item on our list that's necessary for survival is food. Food, items of nutritional value. And we have several different perceptual systems that help us with this task, with finding food. One of those systems is the visual system. And in particular, the ability to see color. So our visual systems allow us to see color, and color helps us with finding food. For one thing, color helps it make it easy to find where the food is. It's easier to see the berries on the left than seeing the berries on the right. Seeing color also helps us determine if the fruit, for example, is ripe, if food is ready to be eaten, if food contains nutritional value. So our color system, our ability to see color, is a huge help and a huge advantage for finding and detecting the ripeness of food. We're able to see color due to the type of receptors we have in our eyes. So this is the retina again. And the first layer of cells are known as photoreceptors. And these are the only neurons that respond to light. And we have two types of photoreceptors. These gray ones are called rods. And rods are what we use to see at night. And then the purple ones are cones. And we have three types of cones. We have a blue cone, a green cone, and a red cone, which refers to the wavelength of light that the cells are most sensitive to. So if we plot that, on the x-axis is wavelength. Humans can only see wavelengths between 400 and 700 nanometers. All other waves are invisible. So when you turn on your microwave, you don't see anything, because we don't have receptors that are sensitive to microwaves. So this is the range of visible light. And wavelength corresponds to what color we see the light as. So shorter wavelengths look blue. Longer wavelengths look red. The y-axis is the cell's absorbance. So how sensitive the cell is to that particular wavelength. The yellow line are the rods, and rods are only sensitive to one wavelength. And then the cones are represented by the blue, green, and red curves to indicate that some cones are most sensitive to blue light, some cones more sensitive to green light, and the other cones most sensitive to red light. And it is through having these three cones that we are able to perceive color. Now, how would we measure color perception? Well, there's a lot of ways to do it, but the way we're going to talk about today is using a method from classic psychophysics. And psychophysics is basically the main method in perception. And the goal of psychophysics is to map the perceptual world onto the physical world. So we're going to simulate being in an experiment. I want you to imagine that you have a button on your left and another button on your right. And we're going to press these buttons in response to the stimuli. And what I'm going to show you is a berry on the left and then another berry on the right. And for whichever one you think is brightest, press that button. So if you see the berry on the right is brighter, press the right button. And if you see the berry on the left is brighter, press the left button. All right, here we go. Even the people in the back need to press the buttons. Get ready. We're going to do lots of these. All right, so you just took part of a classic psychophysics experiment. They have a reputation, and deservedly so, for being the most boring experiments in the history of psychology. But along with being boring, they're actually quite elegant, and you can do a lot of powerful analyses with the responses. This experiment is actually an experiment that I did with students in one of my classes. And what our participants saw was just what you saw. A berry was presented on the left, another one was presented on the right, and then they had to determine which one was brighter. The berry on the left was always the same shade. It's the shade denoted by the arrow here. The berry on the right was one of these 23 shades. All right, and so for some of these shades, it's really easy to tell which one's brighter. But then for some of these shades, it's really difficult to tell which one's brighter. And so we can get a measure of sensitivity. That is, how good are people at differentiating these colors, especially when it's really hard. And what we did was we told some people that they should imagine themselves foraging in the woods, trying to survive, and needing fruit to survive. And we told other participants to imagine being on one of those farms where you can pick your own berries. And what we found was that the people that thought they needed the berries to survive had a heightened perceptual sensitivity. They were better able to determine the differences between these various shades than people that thought they were on a farm or imagined themselves to be on a farm picking berries. So the perceptual system is not just limited by the physiological constraints, but it's also affected by the task at hand. When your task is to do something that's really critical for survival, your perceptual processes operate at a higher level than when you're doing a task that's not relevant for survival. All right, so that's the visual perception's contribution to food. There are also other perceptual systems that help us with food. So if something smells good, that's typically a sign that we want to eat it. If something tastes good, that's a sign that we want to eat it. These two systems are also really useful in detecting poison. Think of what an advantage it is to be able to smell poison before you ingest it. You open up that milk. It doesn't smell good at all. You're not going to drink it. You're protecting yourself from the damage that rotten milk would do to your system. And let's say that something got past your nose. Then you have another backup system with taste. If you put something in your mouth and it tastes bad, you can spit it out without having to ingest it. And in that way, protect yourself from ingesting things that could do harm to your body. Now, of course, there are other things that could do harm to your body besides poison and ingesting things. And for that, we have touch receptors. So the skin is able to detect when different things are touching it or affecting it. And the way that we measure this in the lab is called the two-point discrimination method. And in the two-point discrimination method, a device kind of like this one would be used, where the experimenter varies the length between these 2 points, and then touches some part of your body with either one of these points or both of these points. And the task is to determine whether 1 point is touching you or 2 points. Now, of course, if you have a big enough gap, you're going to be able to tell that it's 2. But the question is, how close can the 2 points be and still detect that it's 2 points? And the results of these experiments is here. The x-axis is a measure of distance between the 2 points. It's hard to see, but that's 10 millimeters, 20 millimeters, 30 millimeters, 40, and 50 millimeters. Pretty small distances. And then the y-axis in this graph is the different parts of the body. Is a lower threshold mean you're more sensitive or less sensitive? Who thinks more? Yeah, it's more sensitive. So a lower threshold, if you're able to tell that it's 2 points, even when they're really close together, that means you're really sensitive at that part of the body. So what part of the bodies are most sensitive? Yeah, the hands, the fingers, your face. And then the parts that are least sensitive are your legs and your torso. So the two-point discrimination method is one way to get at the sensitivity of the different body parts. Another way is to actually look at the distribution of neurons in the brain. How many cells respond to stimuli at each different body part? And what you find is that the number of cells actually corresponds to the sensitivity. So there are a ton of neurons responsible for detecting touch on your fingers and your face. And there are less neurons responsible for these things on your torso and your legs. So if we recreated the body with respect not to physical size, but with respect to sensitivity, this is what it would look like. This is known as a sensory motor homunculus, and the size of the parts are true to sensitivity size, the size of sensitivity, the size of the number of neurons representing that part, rather than to physical size. Isn't that a great graph, so to speak? I mean, it's a great representation of the data. All right, so let's add to our list. We added food and nutrition, we added poison, and we also added other things that could harm or injure the body. The next thing on our list that's important for us to survive are faces. All right, faces are really important, and we have specialized systems dedicated to seeing faces. And these systems are pretty eager. And they lead you to see faces even when they're not there. So some of you may have seen an image like this before where you can find faces. I think there's something like 8 faces in this image. You heard people talk about the man in the mountain, people seeing faces in the rock formations on the mountain, or the man in the moon. But even in these other items, I mean, it's pretty easy to see a face there. You might be able to see a face here. Maybe, if you stretch it there. This system loves faces, and it sees faces in anything that it can possibly see faces. All right, it's a little, like I said, it's a little overeager. Now, how are face perception studied in the lab? Well, in a number of methods. One of them is known as the Margaret Thatcher illusion. And here's a rendition of it. So if you look at these faces, they're not too different. They're pretty similar. But if we turn these faces right side up, the difference becomes obvious. And the one on the right looks much more hideous than the one on the left. These are the same faces as here. But why is the difference so obvious to see here when you can't really tell it here? And the reason is that faces are processed in a manner that's special. And in particular, they're processed holistically. You're processing the face as a whole. And as a whole, it's really easy to see how this doesn't really fit in with the rest of the whole. Whereas when they're upside down, you don't process them holistically. And therefore, the mouth, they look okay on their own. There's nothing wrong with them on their own. The only thing that's wrong with them is how they relate to the other features. But when you see a face upside down, you don't process it holistically. You process the individual features. And as individual features, they're okay. So one way that faces are special is that we process them holistically. We process them as wholes, where we see how the different parts relate. Another way that face perception is studied is using neuroimaging techniques. So in neuroimaging techniques, you measure the oxygen flow in the brain and use that as a measure of activity in the brain. And there's a special area called the FFA, or also known as the face area, that responds to faces. It's the process, the area in the brain that's dedicated to seeing faces. And the way that they discovered this was they put people in the magnet, and they showed them images of faces, images of houses, or blank screens with just a fixation mark in the center. And so they, over time, they showed the fixation mark, faces, and houses. So each of these blocks, the stimulus that was shown, is labeled at the top. And the y-axis is the percent of activity that occurred in that place. The signal changed. And the blue curve corresponds to activity in this special face area. And the red curve corresponds to activity in this area known as the PPA, or also known as the place area. And what you can see is that every time that faces were being shown, activity in the FFA increased, demonstrating that there is a special area in the brain that responds just to faces. Specialized process responsible for seeing faces. Now, seeing faces are important, but there's certain aspects of faces that are also important. And one of these aspects is eye gaze. So you want to know where other people are looking. We're social critters, and when we look at where other people are looking, that provides clues as to what might be important in the world. If I go like this, it's probably a good sign that there's something going on over there that you might also want to check out. The way that this has been studied is using a task known as the spatial cueing paradigm. And in this task, a target's either going to appear in the left box or the right box. So we're going to get out our two buttons again. And when you see the target appear, press the button that corresponds with the box. So if it appears in the right, press your right button. If it appears on the left, press your left button. All right, we're going to start with some practice trials. Good. All right, so now we're going to have a cue. And there's going to be an image of a face appear in the middle. And the person's going to be looking one way or the other. This cue does not predict where the target's going to appear. If the person's looking right, the target has equal chance of appearing in the right or the left. All right, here we go. All right, good. So this task is known as the Posner cueing paradigm, or also known as the spatial cueing paradigm. And any cue can be used in the middle. Often you see people use arrows or some kind of directional stimuli. But then people use this typical paradigm and put faces there with eye gaze, and they manipulated eye gaze. And when the person was looking in the same direction as the target appeared, so when the person looked right and the target appeared on the right, or the person looked left and the target appeared on the left, those are known as congruent trials because the eye gaze was congruent with the target location. And when the person looked at the opposite direction, so if the person was looking right and the target appeared left, or the person was looking left and the target appeared right, those are known as incongruent trials. And what they found, what the researchers who did this found, was that people are much faster in these congruent trials. It's as if when you see somebody looking one way, you can't help but also look that way or orient your attention in that direction. So then when a target also appears there, you find it really quickly. But if a target appears somewhere else, you've already directed your attention to one side, and it takes you longer to then go direct your attention somewhere else. So this is one paradigm that's been used to look at how eye gaze affects our perceptual processes. Now, another item of interest in terms of faces are facial expressions. So we're very expressive, and we convey our emotions often through our faces. And so it's a good thing to be sensitive to these expressions. And the way that this has been studied is using the visual search test. So we've already talked about visual search. Here, the target now, in this case, is an angry face, and the distractors are happy faces. And the one on your right, the target is a sad face, and the distractors are neutral faces. And what you can do with this paradigm is see whether we have certain priorities for certain expressions rather than others. And it turns out we do. We're faster to find an angry face surrounded by happy faces than we are to find a happy face surrounded by angry faces. Which means that we prioritize angry faces, which is probably a good thing, right? If there's something happening in the environment that might cause damage to me, this might lead to an angry face, we want to be aware of that. All right, so let's add faces to our list. We're going to add faces in terms of eye gaze and faces in terms of emotion. And then the next item on our list is location. So knowing where these different things, knowing that these different things are out there doesn't do us as much good unless we know where they are. Right, you need to know what's out there, but you also need to know where is it. So location is a huge thing that's important for us to be able to determine about the environment. And the way that we do this is thanks to the structure of the eye. The structure of the eye is an incredible feat of engineering. It's a very precise piece of equipment. And the goal of the eye is so that for every single object or spot of light out in the world, we want it to project to only a single place on the retina. How many of you wear contacts or glasses? When you're not wearing them, doesn't the world look fuzzy? The reason for that is that when you're not wearing them, a single point in the environment projects to multiple points on the back of your eye. So we have several things that, several features of the structure of the eye that make it so that vision can be precise. For example, we have two lenses. So the cornea bends the light to help focus it. There's also another lens behind the pupil that also helps bend the light to focus it. And one of the reasons why you may need contacts or glasses is that you may have difficulty controlling your lens. Your system may have difficulty controlling your lens to focus the light. And if you don't focus the light, then a single point will project to a range of places on the retina, making things blurry. All right, so we have two lenses to focus the light. We also have lots of supportive structures. So there's a jelly-like substance in the front of the eye known as the aqueous humor, and another jelly-like substance in the middle of the eye known as the vitreous humor. These jelly-like substances help maintain the shape of the eye. If your eye is not the correct shape, so for some of you, the reason you may need contacts or glasses is that your eyeball might be a little too long, or it might be a little too short. If these jelly-like substances do not maintain the shape of the eyeball, then again, a single point of light in the world will project to multiple points on your retina, and things will look blurry. So it's in having these precise mechanics and this precise structure that allows us to detect where exactly things are in the environment in terms of the visual system. Now, the auditory system also helps us detect where things are. Our auditory system is very sensitive to small differences in timing of sounds. So if there's a sound off to the side, like sound B here, it's going to reach the left ear before it reaches the right ear. And that small difference in time helps you localize where exactly that sound is coming from. But for sounds that are straight ahead of you, there's not going to be any difference in the timing. So instead, we have to rely on other cues for sound. And one of these cues is that all these folds in your ear create echoes for the sound. And your system actually uses these echoes to localize sounds. And if you were to basically cut off your ear and cut off all these folds, you wouldn't have access to these echoes. You wouldn't be able to localize sounds unless you could use other cues like which ear heard the sound first. All right, so location is important. The last item on our list are edges. All right, edges are where the important things happen. It's at the edges of objects that we can determine where an object is, what an object is. Edges are a critical feature about the environment that we want to be able to see. And edges are actually something that are extracted in the eye. So this is the retina in the eye. This middle layer of cells help extract these edges. And they extract these edges through a process known as lateral inhibition. And what lateral inhibition does is if a certain cell gets light, it passes on a signal to the next cell, but it passes on a negative signal, an inhibitory signal, to the cells next to it. So where there's light in one place, it's actually going to create an inhibitory process at the neighboring location. And this can be seen with this illusion here. So how many people see this square as being darker than this square? Yeah, most people. This square looks darker than this square. They're actually the exact same shade. But this square is surrounded by a light border. A lot of light is around this border, so there's more inhibition, and it looks darker. Whereas this square is surrounded by a dark border, so there's less light and less inhibition, so it looks lighter. So these are our top 10 features of the environment that your perceptual system is attempting to extract. I'll see you guys on Friday.